This is going to be a talk on BioStruct-Africa. It's a Africa-wide initiative for structural biology. I'm really looking forward to learn more about it. Very welcome, Emmanuel. Over to you.
Thank you. Can you hear me?
Yeah, we do hear you, yes.
You see my screen as well?
And we see it, everything ready to go.
So I want to thank Simon and Lawrence for inviting me to participate in this virtual meeting. I'm always very excited to talk about BioStruct-Africa and make the case for structural biology teaching and research in Africa. That's why I have been traveling across Africa to talk to research institutes about structural biology and about BioStruct-Africa. So a structural biology approach would reveal the molecular basis of disease processes and guide the rational design of new drugs and vaccines. So the usual scenario is to explore a biological space to find a drug target. And then you also explore a chemical space to find a small molecule. And this can be by screening small molecule compound library or using fragment-based drug screening. So in 2016, the first international conference, the first Pan-African conference on crystallography was organized in the University of Chang, Cameroon. So that was when I met my colleague Dauda. And in this meeting, it was pretty evident that structural biology was lacking. So there were very few talks on structural biology, and out of about 100 posters, there were only about 5 of them that was addressing projects related to structural biology. So 1 year after, we established BioStruct-Africa, which is a Swedish registered nonprofit organization. And our mission is to build capacity in structural biology for Africa-based researchers. So the structure of the organization, you have the CEO, the co-founders, board members and members, the partners and scientific advisors. So I was elected as the CEO, and just a brief background about myself. I was born in Cameroon. So I obtained a PhD in Trinity College, Dublin. And after that, I pursued a postdoctoral studies at Stockholm University. And during my time at Stockholm University, I solved the structure of a nucleotide sugar transporter and also developed new tools to facilitate structural biology research. And during this time, I participated in establishing BioStruct-Africa. And then I participated also in a research program that led to the structural determination of a sugar transporter from the malaria parasite, and a couple of other contributions. So one of the co-founders is Dr. Dauda Chauri, and he's based at the synchrotron in Grenoble. And his main role is to facilitate, is to support the users on the Titan Cryos microscope for cryo-electron microscopy. We are actually very blessed to have him with his expertise, so we can tap into his expertise in the future to help us solve structures using cryo-electron microscopy. Then there is Dr. Nji, who is also a Cameroonian, and he's a structural biologist. He did his PhD in Stockholm University, and he's currently a postdoctoral fellow at the Department of Medical Biochemistry and Biophysics at Umeå University in Sweden. So I want to thank our partners. They are institutions and universities based in Africa. So during our workshop, they help us to provide the lab space for us to carry out our demo. Then I really want to commend my members. They've been working really hard to make sure the organization is in good shape. And I really want to thank them for that. So the vision of BioStruct-Africa, firstly, is to train Africa-based scientists in the field of structural biology, to facilitate the relocation of Africans in the diaspora to establish themselves on the continent, to actively promote a vision and roadmap towards building a first African light source, and to form a consortium of African structural biologists working on prevalent diseases on the continent. So in the future, we want to be a multidisciplinary organization with structural biology at its heart. And we also want to establish structural biology centers at African universities. Then in the long run, we would ideally want to have some sort of an African journal of structural biology. I know I've discussed with Lawrence, he has some other ideas, but this is something we all could work towards in the future. So our first workshop was organized took place in Accra, Ghana, and the workshop was structured to have the theoretical part and the practical part. So the theoretical part constitute lectures in structural biology, and then there were some lectures on protein expression, purification, and crystallization. And then there was another lecture to provide an insight into what you can do before and after your beam time to collect X-ray diffraction data. And I really want to thank Diamond Light Source and Professor Tom Blondell for facilitating this process. The next was the hands-on training, in which we, for the first time, actually crystallized lysozyme using traditional vapor diffusion method. As you can see we actually obtained crystals, and you can see there is a smiling face here, very happy. Then we were able to remotely connect to Diamond Light Source and collect X-ray diffraction data. So again, I want to thank Ralph for his help on this. And this is something we can really explore while waiting for the African light source to be built, because rather than traveling to the UK to collect data, you can remotely tap into the synchrotron using a stable connection and your laptop, and then you actually collect your data and process it and get your structure. So after the workshop, we conducted a survey to be able to inform the next workshop what we can do to make it better. And overall, you can see the feedback were very positive, and some recommendations were that we should organize this meeting annually, and also we should increase the length of time of the meeting. So we adopted these recommendations, and we actually organized 2 workshops. We actually planned to organize 2 workshops this year. One was supposed to take place in Kigali, in Rwanda, and the other was supposed to take place in Malawi, which was an invitation by the African Congress of Immunology. But unfortunately, this could not take place because of the COVID-19. So we're really looking forward to the next meeting, and with that, I come to the end of my talk. I'm happy to take some questions.
Okay, well, thank you very much. It's an incredible journey, and really want to say I think it brings the African light source as well one step closer. and there's a remarkable spread in people involved and number of people and so on. And I think it's fantastic. And thank you very much for the presentation. We're still running a little bit over time, so I think we will soldier on, and we will try to pick up questions in the next phase. So if I can call on Marcus Newton, unmute your mic.